Welcome to today's Explainer. We're jumping right into a fascinating and, frankly, a kind of terrifyingly simple corner of biology. Today, we're exploring entities that exist right on the absolute edge of life. We're talking about infectious agents that are so stripped down, they completely blur the line between what's actually living and what's just complex chemistry. So let's break down the science of viruses, viroids, and prions. Okay, to really understand these agents, we first have to ask a fundamental question. Think about the grand tree of life with all its sweeping branches of animals, plants, fungi, and bacteria. Where do these microscopic, infectious agents actually fit in? To fully grasp how they operate, we have to look at how biologists categorize them. Well, the answer is surprisingly elegant. They're considered acellular organisms. You know Robert H. Whittaker's famous biological classification system? It actually left these agents out completely. And the reason why is pretty simple. They lack true cellular structure. They just aren't living cells in the way that a bacterium or a human cell is, and because they're acellular, they completely defy our standard biological classification. Viruses, the crystalline invaders. Let's start our deep dive with the most complex and, honestly, the most famous of our three agents today. The very word virus comes from a Latin root meaning venom or poisonous fluid, which, if you think about it, hits the nail on the head. They have this really fascinating dual nature. As obligate parasites, they essentially wake up, hijack the host's machinery, and replicate, but only once they're inside a living cell. Outside of that host, they are totally inert crystalline structures. They literally just sit there doing absolutely nothing until they bump into a vulnerable target. Now, the detective's story of how he actually found these things took decades. Back in 1892, a scientist named Ivanovsky first recognized certain microbes were causing mosaic disease in tobacco plants. A few years later, in 1898, Bajaranek noticed that the extract from these infected plants could infect healthy ones. He called this mysterious fluid contagium vivum fluidum, a contagious living fluid. But it wasn't until 1935 that Stanley took a massive leap forward. He showed that viruses could actually be crystallized, and he discovered that these crystals consisted largely of proteins. So if they're mostly protein, how exactly are they built? Well, a virus is essentially a nucleoprotein with a highly specific architectural blueprint. First, you have a genetic core of either DNA or RNA, but crucially, it's never both at the same time. Second, this fragile genetic core is surrounded by a protective protein coat called the capsid. And third, this capsid is built out of smaller subunits called capsomeres. These capsomeres arrange themselves into these beautiful, repeating geometric forms to perfectly shield the nucleic acid inside. And here's where it gets wild. Viruses actually customize their genetic payloads depending on their target. If a virus is attacking a plant, it typically uses single-stranded RNA. If it's an animal virus targeting creatures like us, it brings a much more varied toolkit. It could be single or double-stranded RNA or even double-stranded DNA. And then there are bacteriophages, which literally means bacteria infectors. When viruses go after bacteria, their genetic material is usually double-stranded DNA. So the weapon always fits the target perfectly. Viral targets and threats. Let's look at the actual damage these crystalline invaders can do once they breach a host's defenses. The real-world impact here is just massive. For us humans, viruses are responsible for everything from the mild common cold to mumps, herpes, influenza, and historically devastating diseases like smallpox. But they're also an enormous threat to our agriculture. In plants, viral symptoms are incredibly severe. You'll see mosaic formation on the leaves, leaf rolling and curling, yellowing, vein clearing, dwarfing, and stunted growth. They just completely hijack the organism's ability to thrive. Viroids, the naked RNA. This brings us to a really fascinating mystery. We just established that a virus is genetic material wrapped in a protective protein coat. But what if we strip away that shield entirely? Could an infectious agent still exist? Enter the viroid. While a virus has that protective capsid and a nucleoprotein structure, a viroid completely lacks a protein coat. It is physically smaller and consists of absolutely nothing but free, naked RNA. This is a huge deal because it definitively proves that an infectious agent can be far simpler and far smaller than a traditional virus. These things were literally hiding in plain sight until T.O. Diner discovered them back in 1971. This free RNA has a surprisingly low molecular weight compared to the RNA found in viruses. But whatever you do, don't let their microscopic, stripped-down nature fool you. They are highly capable of causing specific, damaging diseases. The most notable is potato spindle tuber disease, which can absolutely wreck agricultural crops. Prions rogue proteins. All right, we've gone from complex viruses to the much simpler viroids. Now, let's take this biological minimalism one step further. What if we strip away the genetic material entirely? No DNA, no RNA. Welcome to the world of prions. These are infectious agents made entirely of abnormally misfolded proteins. That's it. There's no DNA, there's no RNA. It's literally just a protein that has somehow lost its proper three-dimensional shape. It turns into this terrifyingly simple, rogue entity that actually induces other normal proteins in the host to misfold as well. It's like a chain reaction. And because they're just misfolded proteins, they are notoriously difficult to destroy, and the damage they cause is catastrophic. Prions are responsible for severe neurological diseases. In cattle, they cause bovine spongiform encephalopathy, which most of us know as BSE or mad cow disease. In humans, they cause the analogous variant known as CJD. They systematically create these microscopic holes in brain tissue, and they do all of this entirely without the use of genetic code. The threat matrix. Let's pull all of this incredible information together to fully appreciate the scale of how these edge-of-life entities operate. Notice this kind of terrifying pattern of minimalism we've got going on here. We start with a virus, a nucleoprotein combining genetic material and a protein coat, capable of causing diseases like smallpox and influenza. Then, we strip away the protein to get the viroid, just free RNA causing diseases like potato spindle tuber. Finally, we strip away the RNA itself to get the prion, nothing but a misfolded protein causing severe neurological diseases like BSC and CJD. As we strip away parts, these agents become structurally simpler for sure, but they remain incredibly dangerous in entirely different, devastating ways. Which brings us to a final, pretty haunting thought to wrap up today's explainer. If something as staggeringly simple as a single misfolded protein, completely lacking any DNA or RNA whatsoever, can bring down a complex mammal by causing a disease like CJD, what else might be hiding in the microscopic world? We've only relatively recently discovered viroids and prions. The edge of life is this vast, largely uncharted territory, and it clearly still holds profound mysteries. Thank you so much for joining me on this explainer, and keep questioning the invisible world around you.